Hello everyone, this is Nitin Reddy and I'm from DICT, Gandhinagar, India. I'm going to talk about CRISPR and its application in medicine, especially to cure Huntington's disease. So now let's see in brief what CRISPR is and then what Huntington's disease is. So what is CRISPR? To put it simply, CRISPR is a knife to cut the gene, and Huntington's disease is a brain disorder where the affected individual ultimately dies due to malfunction of the brain and nerve cells. Now let's see in detail what Huntington's disease is. Huntington's disease is a brain disorder which causes nerve cells in the brain to break down over the time. Nerve cells are damaged to such an extent, causing various parts of the brain to malfunction and the nerve cells to degrade day by day. There is no treatment as such available with the existing medical practices and available technology. Huntington's disease is caused due to mutated HTT gene, also known as Huntington's gene. Now let's see who is affected and then how it transmits. Huntington's disease in general is an hereditary disease. Affected patient would have one copy of good gene and then one bad copy of the gene. The probability of child being affected by the affected patient is 50%. Let's see how this 50% is being calculated. Every patient affected by Huntington's disease will have one good copy and then one bad copy, as I said earlier. When the child inherits this good copy of the gene, he is safe. He will not show any symptoms of the Huntington's disease. But when the child inherits the bad copy of the gene, he is affected. Let us now look at the symptoms of this disease. People affected with this disease generally show two kinds of symptoms. One is physical symptoms and the other one is emotional symptoms. The most important physical symptom is uncontrolled movements of the body parts. People affected with the disease will have difficulty in terms of controlling their walk, controlling their run, controlling their hand movements, controlling their leg movements, and in general controlling their body parts. People also show this symptom where they find it difficult to speak. By difficulty, I mean there is lack of synchronization between words and thoughts of the affected person. There is another symptom which people show, that is difficulty in swallowing the food. Initially people will not face this problem, but then within course of time people find it difficult to swallow the food. Another symptom which people show up is they tend to discard themselves and then not tend to care for themselves. This is the time when the patient needs care from somebody else. Now let's look at the emotional symptoms. Emotional symptoms are mostly anger, depression, excitement, and then aggression. Most people tend to discard these symptoms because these are in general symptoms which turn up when people are in stress due to their workload or maybe due to their academics or anything else. Let's see what's the actual cause for the disease. The actual cause for the disease is malfunctioning of HTT gene, which is located at the 3' end of the HD gene of the chromosome 4. The affected gene will have higher number of CAG codons. Let's see what are these CAG codons. HTT gene will have repeated trinucleotide CAG codons. Generally, the number of CAG codons are less than 26, but the affected patient will have more than 35 CAG codons. When the protein is encoded or being produced from this gene, it will form a harmful substance because of this excess number of CAG codons in the gene. We've just seen that the excess number of CAG codons are the major cause for Huntington's disease. So let's see how CRISPR and Cas9 could be used to reduce the number of CAG codons to 26 or even lesser than that. Let me explain you in detail what CRISPR is and then Cas9 is. So what is CRISPR and what is Cas9? CRISPR is basically clustered, regularly interspaced short palindromic repeats, which was originally found in bacteria. CRISPR sequences are a crucial component of immune systems of bacteria. Bacteria's immune system uses this to kill virus. Whenever a virus invades a bacteria, it first reads the sequence of the virus, that is a gene sequence of the virus, and then figures out a guide RNA so that it can go and then disable the gene of virus. Cas9 is something which helps bacteria to cleave the gene. In simple terms, this is a knife to cut the gene. Bacteria uses Cas9 either to cleave the gene of the virus in terms of single stranded breaks or double stranded breaks. Guide RNA helps in finding the matching sequence so that you can target the precise location of the gene. How CRISPR could be used? Let's see how CRISPR could be used in terms of Huntington's disease. Huntington's disease is an ideal candidate for CRISPR therapy because the disease is determined by a single gene. This was found by a neuroscientist in Switzerland. We need to stop the production of the protein which is harmful to the brain. To stop the production of the protein, we need to reduce the number of CAG codons in the HTT gene. We can edit the part of the gene where there are higher number of CAG repeats. But to do this, we need efficient guide RNA so that we can go and target the precise location. Cas9 can break the gene, thus halting the production of the dangerous protein. So let's see how the CRISPR could be used to introduce a double-stranded break of the HTT gene. Here if you see, there is this guide RNA which is being used to guide the Cas9 to break the HTT gene. Here if you see, there are excess number of CAG codons which the guide RNA helps in targeting so that it can be broken. So once this excess CAG codons are removed, the number of CAG codons in the HTT gene are less than 26, which is now no more harmful. Scientists have done few set of experiments on mice and then following are the results. The left hand side, if you could see, there are a lot of aggregated proteins, and the right hand side there are no aggregated proteins. Using a virus as a delivery vehicle, the researchers infected two separate groups of healthy adult mice with a mutant Huntington gene, but only one of the group received the therapy. Those without the CRISPR treatment had large areas of protein aggregation, and those with the treatment had almost none. So if you could see here, the left hand side has large set of aggregation, the right hand side there's no aggregation as such. Left hand side is not treated with CRISPR, but then right hand side is treated with the CRISPR therapy. Let's look at some of the issues in the implementation. One of them is blood cells around the brain hinder the movement of the Cas9 nucleus. So when Cas9 is trying to enter the brain cells and then mutate the HTT gene, this blood cells around the brain cells will not allow the Cas9 molecule to do its work. So one of the ways to overcome this is coming up with a special Cas9 molecule attached with GLA peptide, GYR peptide, and then polymer, and then lipids, and then ligand vectors. Another issue in terms of implementation is both the defective HTT gene and then healthy HTT gene might be affected when the Cas9 starts breaking the strands. So what is this? When the guide RNA and Cas9 try to act upon the HTT gene, it will not be able to distinguish between healthy HTT gene and then the mutated HTT gene. In order to differentiate, it's very difficult for the guide RNA. Making efficient and precise guide RNA is again a challenge on which the entire implementation would depend upon, since if you are not able to target a gene precisely, then there is no point in carrying out the editing. So let me conclude with the following things. So one of the major things is choosing efficient guide RNA is a major challenge, solving which will help us carrying out gene editing in a better way. A lot of genetic problems could be solved using CRISPR and its technology. This could be another groundbreaking technology in medicine. There are other issues in terms of medicine where there is no treatment as such with the existing practices or existing medical technology. CRISPR could be one such technology where it could try to explore different ways of solving a problem. Thank you, and this is Nitin Reddy from DISCT, BTech third year. Thank you so much.